Hi everyone, Assalamualaikum warahmatullahi wabarakatuh. Welcome back to Food Technology Study Program, Department of Food Science and Technology, Faculty of Agricultural Engineering and Technology, IPB University. My name is Rati Dewanti Haryadi. I am a professor in food microbiology at IPB University. Today we are going to meet again to discuss ITP 322 on food science and sanitation, and our topic today is on lecture 3, biological hazards and foodborne diseases. Today's topic will cover biological hazard, foodborne diseases, bacterial infection and intoxication by non-spore former bacteria and spore former bacteria, foodborne viruses, protozoa, helminthic parasites, emerging foodborne pathogens, and foodborne disease outbreaks. Upon completion of this lecture, you are expected to be able to differentiate microbiological hazards, describe foodborne diseases, differentiate infection and intoxication, analyze foodborne disease cases due to non-spore-forming bacteria and spore-forming bacteria, and describe foodborne disease outbreaks. Let's start our topic with discussing what constitute biological hazards. Biological hazards that can cause infection consist of bacteria, both non-spore-forming and spore-forming bacteria, viruses, and parasites. That includes protozoa and helminth. The other groups is biological hazard that cause intoxication. That consists of bacteria, both non-spore formers and spore formers, and toxigenic mold. Okay, and I would like you also to understand the terminology commonly used. The first one is foodborne illnesses or foodborne diseases. Those are the scientific terms commonly used to describe food infection and or intoxication. Meanwhile, food poisoning is a common term to describe intoxication or poisoning caused by either microbial toxins or chemical toxic compounds. Okay, so what is bacterial infection? Bacterial infection usually starts with viable or living bacterial cells entering our body through the food that we consume. Once they enter the body, they may be able to establish themselves, this is called colonization, at a certain site, for example, on the intestines, and they may penetrate to the bloodstream, this is called bacteremia or septicemia, and they may also colonize other organs such as liver, kidney, etc. This part is called invasion. They may produce toxins within the human body, and this is called toxico-infection. Usually, bacterial infection has a slow onset. That means there is a long period of time between the time people consume food and the time the symptoms start to occur. An example for this is cholera due to Vibrio cholerae, salmonellosis by Salmonella, and typhoid fever by Salmonella typhi. Foodborne infection can also be due to viruses. In this case, viruses enter human body through the food ingestion, and inside the host, the virus will recognize receptors on the host cells, attach, insert their DNA or RNA, which will eventually recombine with the host cell's DNA. The virus then replicates within the host cells, and when the host cells are producing the viruses in large number, they may eventually die. We all know already now that both bacteria and viruses could cause illnesses. Now, what about their fate in foods? Bacteria grow and multiply in foods, while viruses as well as protozoa do not grow or multiply in foods because they are obligate intracellular parasites. That means they have to be living inside a living host to be able to replicate. The stability of viruses in foods are depending on the strain types and also temperature. Generally, viruses are more stable at refrigeration temperature. However, they are easily killed by heat pasteurization, while some bacteria could produce spores, thus become heat resistant. Virus and protozoa do not produce toxins, that's why they cannot cause intoxication, while some bacteria do. Let's move to bacterial intoxication. Bacterial intoxication started when microorganisms in food form toxin. The foods containing toxins are then consumed by humans, and by this time, the bacteria themselves may no longer be viable in the food. The toxins that are consumed could bind to receptors in humans and cause illnesses. The time between consumption and the appearance of the symptoms are generally quick. That's why it's called rapid onset. Examples for foodborne intoxication are Staphylococcus poisoning by Staph aureus enterotoxins and adult botulism by neurotoxins produced by Clostridium botulinum. Now, some intoxication are not caused by bacteria, that is called non-bacterial intoxication, and we have learned some of this in lecture 2. Some intoxication could be caused by mycotoxins, that is toxin produced by mold, toxin produced by algae, or natural toxins from plants. Intoxication can also be caused by inorganic chemical contaminants such as heavy metal, pesticides, etc. And unlike bacterial toxins, the above toxins are not protein, thus they are heat resistant and cannot be inactivated by temperatures commonly employed in food processing. Meanwhile, bacterial toxins are generally extracellular proteins and they are heat sensitive. There are some exceptions though. Staph aureus enterotoxins is a heat-stable protein toxin, and bronchotoxins by Burkholderia gladioli is also heat resistant. Previously, we have learned that some bacteria are capable of producing spores and called as spore-forming bacteria. And these bacteria are more resistant to heat. Meanwhile, some bacteria that are not capable of making spores or non-spore formers can easily be killed by temperatures under 100 degrees. So people can get illnesses from non-spore forming bacteria when they are eating unprocessed food because this unprocessed food contain the bacteria, or they eat processed food that get contamination post-processing, or they got illness from food that are underprocessed or not processed well. Meanwhile, spore-forming bacteria could cause illnesses usually in processed or heat-treated foods. During the heating, the bacteria could form spores, and when the heating is not sufficient, these residual spores may germinate during slow cooling and grow because specific conditions exist. For example, absence or presence of oxygen, a certain pH, or time. The knowledge on the spore-forming bacteria are the basis for 12D process in commercial sterilization. This following tables shows you non-spore-forming bacteria that can cause infection. So Salmonella, Shigella, Vibrio cholerae, and Vibrio parahemolyticus are all non-spore-forming, that's why they are heat sensitive. However, Salmonella has a unique characteristic that is dry resistant. It could cause illness from gastroenteritis up to typhoid fever. The outbreaks linked to Salmonella has been caused by various food, like you see here, from undercooked beef, poultry, raw eggs, up to produces, peanut butter, nuts, and paprika sprinkled chips. Shigella is usually related to human fecal and usually contaminated food through poor hygiene. This bacteria cause dysentery and usually linked to raw foods. Vibrio cholerae could cause cholera. It has been linked to human feces but also brackish water. So in addition to fecal contaminated water, outbreak has been reported with raw vegetables and raw seafoods. Vibrio parahemolyticus also cause gastroenteritis. It is associated with the brackish water, thus usually is linked to the consumption of raw seafoods, sushi, or sashimi. This list also shows non-spore-forming bacteria that could cause infection. All four are the groups of E. coli that has different characteristics. The first one, enteropathogenic E. coli or EPEC, is capable of adhering to intestine, causing infant diarrhea, and generally related to water or milk. Enterotoxigenic E. coli can produce toxins. It causes similar illnesses and also associated with contaminated water or milk. So the third one is enteroinvasive E. coli, which has a characteristic similar to Shigella. It causes dysentery, and similar to the other two E. coli, they all are related with water and milk. The fourth group are more food-related. This group of E. coli is called enterohemorrhagic E. coli or EHEC. EHEC own cigatoxin gene, and they can cause severe illnesses such as bloody diarrhea, kidney failure, and thrombotic thrombocytopenic purpura. The source of bacteria is on human and also cattle, and the outbreaks related to EHEC has been linked to hamburger, sausages, up to sprouts. This table still deals with non-spore-forming bacteria that can cause infection. The first one is Campylobacter jejuni that can live only when oxygen availability is low. It's called microaerophilic. It also has a property of capnophilic, that means it needs 5 to 10% of carbon dioxide to grow. It causes enteritis, diarrhea, and sometimes bloody diarrhea, and the main source is poultry. That's why the outbreaks are generally associated with undercooked poultry. Yersinia enterocolitica and Listeria monocytogenes both has a similar property, that is psychrotrophic. That means they can grow at refrigeration temperature. Yersinia enterocolitica has been associated with pig and wild animals and cause outbreaks through the consumption of pork, seafoods, and pasteurized milk. Meanwhile, Listeria monocytogenes cause a lot more severe illnesses because it can be transferred to fetuses. So it causes only flu-like symptoms in adults, but in pregnant women it can cause miscarriage, stillbirth, and meningitis. Several outbreaks have been reported with Listeria monocytogenes, coleslaw, raw milk, soft cheese, caramel apple, and enoki mushroom have been associated with Listeria monocytogenes outbreaks. Now let's move to non-spore-forming bacteria that can cause intoxication. The first one is Staph aureus. We have talked about it before. It produces heat-stable enterotoxins, but it also a poor competitors. That's why most of the outbreaks are due to processed food and not raw foods. The illnesses due to these enterotoxins is generally nausea and vomiting, and the bacteria are generally coming from the workers because it is usually originated from nose and throat. The other bacteria that can cause intoxication is Burkholderia gladioli. They produce toxoflavin and bone crack toxin. Both are neurotoxins that attack nerve system. The toxin accumulated in the body can cause death, and this toxin has been exclusively associated only with tempe bongkrek, that is a fermented food made from coconut waste, and is a very local, specific food in the Central Java, South Central Java. Let's meet spore formers that can cause infection and/or intoxication. The first one is Clostridium perfringens. The spore-forming bacteria are also known as pathogen. It causes infection and usually associated with the consumption of cooked beef or gravy. Bacillus cereus can produce both diarrheal and emetic toxin that can cause vomiting. The bacteria can cause intoxication due to the emetic toxin, but also cause infection due to the diarrheal toxin. Cereals, rice pudding, and fried rice has been associated with outbreaks of Bacillus cereus. Clostridium botulinum is unique because it is obligately anaerobe, that means can only grow in the absence of oxygen. It also produces heat-sensitive neurotoxins called botulin that cause paralysis. Intoxication due to botulin is called adult botulism, usually characterized by paralysis, respiratory failure, and may cause death. The bacteria can be found easily in soil, sediment, and gastrointestinal tract of animals, including fish. Several food processed with canning, with insufficient heating, has been reported to cause outbreaks such as canned pea, canned bamboo shoots, smoked fish, potato salads, and garlic in oil, as well as carrot juice. Clostridium botulinum also has been reported to cause infection that is called infant botulism in babies, and the food associated with this infection is honey. Okay, let's change our subject to viruses that can also cause illnesses in humans. Viruses is also sometimes known as particle with the size of smaller than bacteria. Viruses contain only DNA or RNA and protein and sometimes lipid. Foodborne viruses, usually a single-stranded RNA viruses, having a coat protein and enclosed in a lipid material originated from the host cytoplasmic membrane. Viruses are often blamed for illnesses that do not respond to treatment. Virus contamination in food most often is resulted from human carelessness. The risk of virus contamination via food is greater for food that are handled intimately and not cooked thereafter. There are three viruses, foodborne viruses, that I would like you to learn. The first one is hepatitis A viruses that cause hepatitis. It is usually transmitted through anal-oral routes. The outbreaks associated with hepatitis A viruses is usually associated with shellfish, but it can also be associated with water as well as any food handled with contaminated water. The other viruses are rotavirus, which are mostly waterborne, and then Norwalk-like virus that can cause stomach flu. The first isolation of Norwalk-like virus is in the patient with infant diarrhea in Norwalk, Ohio. Next is protozoa. They can also cause illnesses. Protozoa are single-celled animals that derive their livelihood entirely from the host, for example, human. They carry out sexual reproduction, produce cysts that are shed in the feces and can contaminate the environment, and the cysts are generally more resistant. They cannot multiply outside the host, including in foods. The general preventions for protozoa are proper disposal of feces, wastewater treated properly before discharge, proper hand washing after defecation, filtration, because generally cysts are more resistant to chlorine, cooking kills the cyst, and boiling makes water safe. There are several protozoa important in foods. I would like you to only know the examples, three of them. The first one is Amoeba histolytica. It causes amoebic dysentery. The hosts of these protozoa are human, dogs, and cats, and they are particularly important in tropical regions. The prevention for Amoeba histolytica illnesses is personal hygiene and the use of safe water. Giardia lamblia is also mostly waterborne and can infect the intestine. Meanwhile, Toxoplasma gondii are parasites in the brain, heart, and muscle of cat, lambs, etc. The prevention from Toxoplasma gondii include cooking the beef and lamb thoroughly. Let's move to larger parasites. The first one is roundworm. And one example of important roundworm is Trichinella spiralis that is usually form larvae inside the pig's muscle. To inactivate roundworm such as Trichinella, you have to cook thoroughly. Usually at a commercial setup, you will apply 58 degrees Celsius, and in household, you use 66 degrees Celsius. Beef is roasted until no longer pink at 77 degrees Celsius or freeze at minus 15 degrees Celsius for 20 days. Other roundworms include Ascaris lumbricoides, that is usually needs a proper disposal of domestic waste because the source is feces, and Anisakis. Anisakis is usually or has a definitive host of dolphin. However, it may be found in ikan kembung and mackerel and any fish that ingest the larvae. Another form of parasites are tapeworm, Taenia saginata, or in the larval stage is called Cysticercus bovis, is found in cow. Healthy people can be infected and become asymptomatic. The proper prevention is disposal of domestic waste, inspection of beef muscle, and cooking to a minimum of 57 degrees Celsius or freezing at minus 50 degrees Celsius for 5 days. Flukes is another form of parasite worm, and Fasciola hepatica is usually found in the liver of cow. In the past few years, people found new pathogens or old pathogens that behave differently or cause a different kind of illnesses. This group of pathogens are classified as emerging foodborne pathogens. There are several classifications of emerging foodborne pathogens. The first one are pathogens whose incidence increases in the past 20 years and are anticipated to increase in the future. Second group are pathogens that experiencing genetic mutation and become more virulent. The third one is pathogen that moves to new geographical area. The fourth one is food pathogen with the new modes of transmission. And then the last group is pathogens on the watch. Why do pathogens emerge? There are several studies on the emerging pathogens, but it can be simplified. There are three factors that support the emerging of pathogen. The first one is external factors, constitute of globalization of food supply, introduction of pathogen to new geographical area, heavy migration, movement of people, such as tourists, refugees, changes in lifestyle, development in food processing technology, as well as climate change. The second factors is host factors, changes in human demography now. There are more old people, and also the persistence of malnutrition and presence of HIV patients. The third factors come from the microorganism itself. There are changes in microbial population, such as genetic transfers, and also the development in detection technique that allow us to detect pathogens that probably previously are not detectable. Let's start with the first group of emerging pathogens. Neuroviruses, previously known as non-walk-like viruses, is now reported as the major cause of foodborne diseases in the United States. This virus has long history association with gastroenteritis. However, the incidence of noroviruses has increased lately, probably due to several factors such as consumption of more fresh fruits, traveling in extended period of time, such as in a cruise ship, improvement in the detection method for detecting the viruses. Several outbreaks associated with the consumption of raspberry and cruise ship due to noroviruses from the infected chef, for example, has been reported. An example for the second group of foodborne pathogen is EHEC or Enterohemorrhagic E. coli or STEC, as well as VTEC. This group of E. coli is known to be resistant to freezing and acid, and although it has been isolated previously, the first time this bacteria is reported as foodborne pathogen was in 1982, and the bacteria involved in the outbreak is E. coli 015787. I have mentioned previously that this bacteria caused illnesses more severe than any other E. coli, and the most recent outbreaks occurred in Germany in 2010 that involved 4,000 illnesses with 50 deaths due to E. coli 0104H4. It is now known that E. coli 0157H7 is E. coli that STX toxin gene from Shigella dysenteriae, while E. coli 0104H4 is E. coli 0157H7 that gets gene from anaeroaggregative E. coli. The third group of emerging pathogen involved with pathogen that move to a new geographical area. In this case, one example is the moving of Cyclospora to North America through several produce that presumably imported, such as raspberry, Thai basil, cilantro, and snow peas from other countries. An example for group 4 is Salmonella enteritidis phage type 4 or PT4. As you all know, that Salmonella enteritidis has always been associated with poultry and eggs, especially cracked eggs that allow this bacteria to enter the egg. However, certain Salmonella enteritidis was suddenly found in outbreaks related to grade A or uncracked eggs. Apparently, the bacteria resides within the egg yolk because the hen's ovary was infected. Thus, the terminology trans-ovary transmission occur. Food associated with outbreaks include runny, scrambled egg from pulled egg, as well as eggnogs. The last group of the emerging pathogen are pathogen on the watch, which consists many examples. However, I would only introduce you to one example, that is Chronobacter spp. This bacterium used to be known as Enterobacter sakazakii, and it's a foodborne disease mainly associated with the consumption of powder infant formula. This bacteria has been reported to cause meningitis, especially in preterm babies, low birth weight babies, and although the incidence is low, but the fatality is pretty high. In our group in IBB University, we have worked with this emerging pathogen, and we have isolated the bacterium from powdered infant formula, follow-on formula, weaning food, and other types of foods. We also reported that most of the isolates are genetically related. This bacterium can be inactivated to undetectable level with a reconstitution at 70 degrees Celsius. However, they could grow during hang time at room temperature. The bacteria survive better at 50% of relative humidity and survive dry condition, and they could enter viable but non-culturable state, that is, not growing on agar but still alive, upon dry and acid stresses. That sums up our discussion on foodborne pathogen. Let's discuss a little bit about foodborne disease outbreaks. Foodborne disease outbreaks is an event in which two or more people becoming ill with similar symptoms after consuming the same foods. People involved in the outbreaks is called cases, and generally foodborne disease outbreak worldwide is underreported. In Indonesia, we call outbreak as kejadian luar biasa, and to be able to find the agent that cause outbreaks, it requires thorough investigation, and the science that do this is called epidemiology. This is the top foodborne pathogen that cause foodborne diseases in Southeast Asia, according to WHO. You can see Campylobacter as the one that causes the most outbreaks. However, when we rank this pathogen based on the ability to cause death, then Salmonella typhi rank first and Campylobacter rank the last one. Here are summaries of pathogen causing diarrhea in Indonesia. Salmonella, Shigella, Vibrio cholerae, and pathogenic Ecoli are important foodborne pathogens in Southeast Asia, including Indonesia. We do not have data on real outbreaks, but the isolation frequency from fecal of diarrheal patient in Indonesia is predominantly Vibrio cholerae, 37%, followed by Shigella flexneri, 27%, Salmonella, 17%, EHEC, 1%, and Norwalk-like virus or norovirus, 1%. Parasites, protozoa, are commonly isolated from children above 30 years old, but surveillance for foodborne diseases is not regulated specifically. We only can see surveillance for diarrhea, hepatitis, as well as typhoid fever. This is the end of my presentation. I hope you learned about biological or microbiological hazard as well as foodborne diseases. See you next time. Assalamualaikum warahmatullahi wabarakatuh.